Today we will talk about design and potential applications of nanoparticles in medical imaging. This video will introduce the nanoparticles used as contrast agents in medical imaging. So what is contrast agent? Contrast agent is injected into the bloodstream and circulated through our body. Although contrast agents can be quickly removed via our kidney, it will contain in your examination. It greatly improves the quality of images we obtain. There are some conditions such as infections and tumors, which are not visible without the presence of contrast agent. Also, there are structures like arteries and veins, which are much more readily visible with the presence of contrast agent and allow us to separate out the adjacent anatomy. Recently, studies have started to aim on using nanoparticles contrast agent to improve the precision of diagnostic medical imaging. But what is nanoparticle? According to the International Union of Pure and Applied Chemistry, a particle of any shape with the dimension in the range of 1 nanometer to 100 nanometers can be identified as a nanoparticle. So why are nanoparticles important? Because they show very different physical and chemical properties, such as optical, electrical, mechanical, magnetic, and catalytic properties. When the light interacts with bulk materials, the amount of reflected, scattered, absorbed, and transmitted light decides the color we see, and is unique for the material itself. Different sizes and shapes of nanoparticles of the same material show different physical and chemical properties. The technology is based upon contrast nanoparticles with the properties of cell targeting. The ability of targeting for nanoparticles is to identify tissues of interest and selectively accumulate within it. There are two ways for nanoparticles contrast agent to target the candidate tissues, which are passive targeting and active targeting. Passive targeting relies on the various physiological abnormalities or phenomena that are present in a disease condition in order to provide selective accumulation. The well-known example is the enhanced permeability and retention effect seen in solid tumors. On the other hand, for active targeting, nanoparticles without the ability to directly target the candidate tissues can be functionalized with a biological targeting agent, such as an antibody. For the detection of cardiovascular disease, contrast nanoparticles are introduced into the bloodstream. In this example, the particles are targeted towards proteins on the surface of an atherosclerotic plaque. When the particles bind to the plaque, high loading of the particles can be achieved due to their small size. After injecting the antibody nanoparticles contrast agent, anti-nanoparticles can be clearly detected during the imaging process. Successful experiments in vivo have demonstrated that nanoparticles can use in cell tracking due to its high sensitivity. The key factor for effective contrast agent is use of materials with high atomic numbers. The higher the atomic number of the contrast agent, the better the resultant contrast. Nanoparticles with higher atomic number can induce strong attenuation and be ideal candidates as contrast agents. According to the study by Bonatelli Bass and his research team, the concept of cell tracking with nanoparticles enables non-invasive tracking and visualizing of the therapeutic cells as they migrate and accumulate at the site of injury and malignancy. The antibodies were conjugated nanoparticles in vitro, then intravenously injected to the subject. In vivo, CT imaging enables real-time, non-invasive cell tracking. Contrast agent was injected via tail vein and reads the image continuously for 30 seconds during and after injection. Using this imaging method, it was possible to visualize contrast as it moved from the vena cava to the heart before distribution to the arterial system. Due to the result, The clinical CT scanner provided excellent temporal resolution as tantalum oxide nanoparticles clear from the blood within seconds following injection. Given that the vena cava and abdominal aorta in the rat are approximately 1 to 2 mm, the 3D rendering of in vivo imaging exam on rats was performed to compare the effects before and after the injection of nanoparticles contrast agent. Take another study from Mir as an example. Different perspective was shown in cell tracking use gold nanoparticles as contrast agent in CT imaging. The use of gold nanoparticles can examine the distribution, migration, and kinetics of T cell at the tumor site with the whole body CT imaging. The other practical example would be superparamagnetic iron oxide nanoparticles plots for molecular imaging. Superparamagnetic iron oxide nanoparticles have demonstrated their utility as an important tool for enhancing magnetic resonance contrast. They allow researchers to monitor not only anatomical change, but physiological and molecular change as well. Compared to typical contrast agent, there are many advantages of using nanoparticles contrast agent in medical imaging. Nanoparticles possesses typical contrast agents on many aspects, such as overall cost, labeling efficiency, sensitivity, and low side effects. Thank you for watching this video.